Okay, guys, so we're done with the first integrating center. We're done with the first region that is responsible for integrating and processing, controlling, receiving, and sending out a motor output. That's your spinal cord. Now we're going to look at the other computer, and that is your brain. So the brain is, in order to understand it properly, we have to align our directional terms, our laterality. What is rostral? Rostral meaning is closer to the nose, is closer to the forehead. And there's also caudal. There's also caudal. So caudal is toward the spinal cord. Caudal is toward the spinal cord. So there are major portions of the brain. There's the forebrain, the cerebellum, and the brain stem. These are the major portions of the brain. The cerebrum is the largest part of the forebrain, and it's about 83% of your brain volume. It is made up of your cerebral hemispheres, and these are a pair of half globes of the cerebrum. These are a pair of half globes of the cerebrum, and there are gyri. These gyri are thick folds on the cerebrum surface. These gyri, these are the folds, but each fold is sandwiched between shallow grooves known as the sulci. These sulci are shallow grooves between the gyri. There is a longitudinal cerebral fissure. This longitudinal cerebral fissure is a deep groove that separates the cerebral hemispheres. It is made up of the corpus callosum. This corpus callosum is a thick nerve bundle at the bottom, at the base of the longitudinal fissure that connects the hemispheres. So there are major brain regions known as the cerebellum. The cerebellum is the second largest part of your brain. It is located in the posterior cranial fossa, and it is separated from the cerebrum by a transverse cerebral fissure. And it also contains fissures and sulci and gyri. So the brainstem is known as the rest of the brain, which is made up of the pons, as we drew earlier, the pons, the medulla, and the midbrain. So these are our sulcus. These are our sulci, our cuts, and between them, these folds are known as the gyri. Gyri, not gyro. So these gyri, they are sandwiched between the grooves, and they represent, very important, that is how your centers are made, in the form of a gyri. Okay, so you have the corpus callosum. This is at the base of the brain, and this is the midbrain and the pons, and that's your medulla. You see your midbrain, your pons, and the medulla. Those three, the midbrain, the pons, and the medulla, are known as what? Brainstem. They are made up of the brainstem. The choroid plexus is very important. Why is the choroid plexus is very important? The choroid plexus is very important because that is where we filter blood plasma. You know, we said we don't make the cerebrospinal fluid in Arizona. We make it inside the capillaries of the brain in the choroid plexus. So we filter the blood plasma and we add some substance to it. So cerebrospinal fluid is not, if you examine it, take a sample of cerebrospinal fluid and plasma fluid, and you examine both of them, you're really not gonna find that they are identical. So there are some minor modifications, but for the most part, they're really simple. They're really similar. Embryonic development. So there is so the nervous system develops from the ectoderm, which is the outermost tissue layer of the embryo. And there is neurulation. The neurulation occurs within the first 3 weeks. And there is within the first 3 weeks, and the dorsal midline of the embryo thickens to form the neural plate. So that shows us the formation of the embryonic neural tube. This neural tube and the neural crest is the basis for the embryonic development that leads to the genesis and the making of the nervous system. So you have the neural crest. The neural crest is a longitudinal column on each side of the neural tube that is formed from ectodermal cells. It gives rise to the two inner meninges, most of the peripheral nervous system, and other structures of the skeletal, integumentary, and endocrine systems. By the 4th week, the neural tube exhibits 3 primary vesicles at its anterior end. You have the forebrain, that is known as the prosencephalon. The forebrain is known as the prosencephalon. And you have the midbrain, that is known as the mesencephalon. The midbrain is known as the mesencephalon. And you have the hindbrain. The hindbrain is known as the rhombencephalon. The rhombencephalon. So the nervous system development continues, and by the 5th week, the neural tube subdivides into 5 secondary vesicles. So the forebrain divides into 2 vesicles, the telencephalon and the diencephalon. The midbrain remains undivided and retains the name the mesencephalon. The hindbrain divides into 2 vesicles, the metencephalon and the myelencephalon. So these meninges are very important because they are made up of 3 membranes. So 3 membranes surrounding the brain and the spinal cord. They lie between the nervous tissue and the bone. And they protect, they encapsulate, and they surround our nervous system. And they are made up, as we said, from 3 different layers. You have the dura mater and the arachnoid mater and the pia mater. The dura mater is a very tough, rigid, strong layer to protect the brain from trauma. The arachnoid mater is very important because right beneath your arachnoid mater is your cerebrospinal fluid between the arachnoid mater and the pia mater, and that is going to secure. So the arachnoid mater is going to secure for me between the pia mater and the brain. That's going to secure for you the soft, visceral brain. So in order to be in touch with the brain, you have to be as soft and as delicate as the brain. And that's why the pia mater is the softest. And where do we find our cerebrospinal fluid? It is found between the arachnoid mater and pia mater. Very good. Okay, so these sinuses, even in the respiratory system, you have nasal sinus. The nasal sinuses, these are spaces located where the periosteal and the meningeal layers are separated. So you have air in your nasal sinuses, and in the brain you have this very important dural sinuses that have your blood circulating through the brain. So we talked about these. So meningitis is when bacteria, viruses, fungi, and these infectious microbes leak through your nose or leak through your mouth and enter into these dural sinuses. And then, like opportunistic parasites, they take an opportunity to enter into your cerebrospinal fluid. And once they are there, they are really hard to get. So, this cerebrospinal fluid and the blood-brain barrier, this brain barrier system is very strong that patients who have brain cancer, God forbid, they have a very difficult time being treated by the chemotherapy because the chemotherapy does not make its way to the brain tissue, does not make its way to the brain tissue due to this brain barrier, due to this brain barrier. So ventricles, there are four internal fluid-filled chambers of the brain. There are two lateral ventricles, one in each cerebral hemisphere. There is a third ventricle that is narrow, medial space between beneath the corpus callosum, and there is a fourth ventricle that is a small triangular chamber between the pons and the cerebellum. So the ventricles are connected through this interventricular foramen, and we have cerebral aqueduct. So these are your two lateral ventricles where the cerebrospinal fluid is made. Then through the interventricular foramen, this cerebrospinal fluid in your lateral ventricle is going to pass into the third ventricle. Then from the third ventricle, you have the cerebrospinal fluid that is going to flow through the cerebral aqueduct is going to flow into your, you see, this fourth ventricle. See the fourth ventricle all the way in the bottom. Third ventricle is in the middle, and the lateral ventricle, both of them are at the top. That's an excellent model. So now we see all these ventricles. One, two. So these are your lateral ventricles. In the middle is your third ventricle, the intervertebral foramen. That is how the cerebrospinal fluid is flushed down from the lateral ventricles into the third ventricle, and from the third ventricle into the fourth ventricle, you have your cerebral aqueduct. So cerebrospinal fluid is clear, colorless liquid that fills the ventricles, which are canals of the central nervous system, and beneath it bathes the external surface of your brain. So this production of the cerebrospinal fluid begins with, remember, blood plasma. That is the raw material from which we make our cerebrospinal fluid. We make it from the blood, not the red blood cells, not the white blood cells, but the plasma. We filter the plasma, and from that we homogenize a mixture and with different solutes to make your cerebrospinal fluid. And a choroid plexus. The choroid plexus is a spongy mass of blood capillaries on the floor of each ventricle. So there are ependymal cells. These ependymal cells are neuroglia that line the ventricles and cover the choroid plexus. There are ependymal cells that modify the filtrate. And compared to the plasma, the cerebrospinal fluid has more sodium and chloride. So there's less potassium, calcium, glucose, and very little protein. So the cerebrospinal fluid continuously flows through the central nervous system. It is driven by its own pressure, the beating of the ependymal cilia, and the pulsations of the brain. So your heart, when your heart is beating, this vibration is causing the shaking of your brain to move this cerebrospinal fluid. This path is very important of how we get your cerebrospinal fluid. You need to know that. So the cerebrospinal fluid is secreted in the lateral ventricles. And through the intervertebral foramina, it is secreted into the third ventricle. So down the cerebral aqueduct into the fourth ventricle. You have third and fourth ventricles that add more cerebrospinal fluid along the way. So the flow of the cerebrospinal fluid continues, and all cerebrospinal fluid is ultimately going to escape through three pores that lead into the subarachnoid space of the brain and the spinal cord surface. There's a median aperture and two lateral apertures. The cerebrospinal fluid is reabsorbed by the arachnoid granulations, and these arachnoid granulations, these are cauliflower-shaped extensions of the arachnoid meninx. So this figure here, the flow of the cerebrospinal fluid, that is very important. Where you have the cerebrospinal fluid is secreted by the choroid plexus. This choroid plexus makes the cerebrospinal fluid from what? What do we make the cerebrospinal fluid from? Blood plasma. Excellent. And then the cerebrospinal fluid that is made in the choroid plexus of the lateral ventricle. This cerebrospinal fluid is going to flow through the interventricular foramina into the third ventricle. Then the choroid plexus in the third ventricle is going to add more cerebrospinal fluid. It's going to add more cerebrospinal fluid, and the cerebrospinal fluid is going to flow down the cerebral aqueduct to the fourth ventricle. From the fourth ventricle, the choroid plexus within this fourth ventricle is going to add more cerebrospinal fluid. And the cerebrospinal fluid is going to flow out two lateral apertures and one median aperture. The cerebrospinal fluid is going to fill the subarachnoid space and is going to bathe the external surfaces of the brain and the spinal cord. So at the arachnoid granulations, the cerebrospinal fluid is going to be reabsorbed into the venous blood of the dural venous sinuses. So why do we need? So now we learned how do we make the cerebrospinal fluid from the blood. There's the raw material in the blood plasma in the choroid plexus stored in the ventricles, and the ventricles keep flushing it from the lateral ventricles through the intervertebral foramen into your third ventricle, and then through the cerebral aqueduct into your fourth ventricle. Now, why do we need it? Why do we go through all this hassle of making the cerebrospinal fluid? Number one is the buoyancy. We need our brain to be floating in our cranial cavity, and that allows the brain to attain considerable size without being impaired by its own weight. So if the brain rests heavily on the floor of the cranium, the pressure would kill the nervous tissue. So protection. So the cerebrospinal fluid is important for protection, and it protects the brain from striking the cranium when the head is jolted. So shaken child syndrome and concussions occur when there is so much severe trauma, and the shaking that's going to cause the cerebrospinal fluid is able to accommodate and perform like a cushion, but within limit, within reasonable jolting and jostling and trauma. Not very severe. You know, this boxing and getting punched in the head for money. Okay, and getting slapped in the head too. That's a new one now. Getting slapped in the head for money. So now you're going to destroy your brain centers for $500. Okay, so chemical stability. So the flow of the cerebrospinal fluid rinses away the metabolic waste from the nervous tissue and homeostatically regulate the chemical environment. So now we need to nourish and perfuse our blood, our brain with oxygen. Why is blood supply very important to the brain? So we need oxygen for oxidative phosphorylation and for aerobic respiration. We need that one. We also need the blood supply in order to provide our brain tissue with glucose, with carbohydrates, in order to get ATP metabolism, in order to metabolize carbohydrates to get ATP. What's the name of the pathway that the cell utilizes to get ATP from carbohydrates, glucose? What is that called? Glycolysis. What's the name of the metabolic pathway that your cell uses in order to get ATP from fats and lipids? Beta oxidation. Beta oxidation is the metabolic pathway that your cell uses to get ATP out of lipids. What's the name of the pathway that your cells use in order to get ATP from proteins? Protein catabolism. So you know you have pesos, you have pounds, you got to convert that to dollar. So that's what metabolism is, exchanging, breaking the bonds on all of these macromolecules to get energy. So the brain barrier system is important because it regulates what substances can get from the bloodstream into the tissue fluid of the brain. So although the blood is crucial, it can also contain harmful agents. So the brain barrier system is selectively permeable. It is highly permeable to water, glucose, and lipid-soluble substances, such as oxygen, carbon dioxide, alcohol, caffeine, and nicotine anesthetics. It is slightly permeable to sodium, potassium, chloride, waste products such as urea and creatinine. 2 points of entry must be guarded. There is the blood capillaries throughout the brain tissue, guarded by the blood-brain barrier. And capillaries of the choroid plexus. These are guarded by the blood cerebrospinal fluid. So the blood-brain barrier protects the brain at the blood capillaries. They consist of tight junctions between endothelial cells that form the capillary walls. So during the development, the astrocytes reach out and contact the capillaries with their perivascular feet. So they induce endothelial cells to form impenetrable tight junctions. So anything leaving the blood must pass through the cells and not the gaps between them. So the endothelial cells can be selective, unlike the simple gaps. And they exclude harmful substances, but they allow necessary ones to pass into the brain tissue. So the blood cerebrospinal fluid barrier, that protects the brain at the choroid plexus. These are formed by tight junctions between the ependymal cells, and these tight junctions are absent from the ependymal cells of elsewhere. Okay, the brain barrier system can be an obstacle, we said, for antibiotics and the cancer drugs. So the chemotherapy is not going to be very effective, particularly for brain cancer, due to this brain barrier system that is standing in the way between the chemotherapy and the tissue. So we said we have to have a very good concept of what is a stroke. So stroke is a cerebrovascular accident. It is a cerebrovascular accident, represents sudden death of the brain tissue due to interruption of blood supply. It can occur at any age, but two-thirds are over 65 years old. So what are the differences between hemorrhagic and ischemic stroke? So in hemorrhagic stroke, in the end of the day, if you want to know the difference between two things, you always have to agree on the similarities. So what are the similarities? What are the similarities between the what are the similarities between the hemorrhagic and ischemic stroke? Sorry? They are both disruption of blood flow to the liver, to the brain. So in the end of the day, I'm supposed to get 100% of blood arriving to all of my, this is my tissue. All these dots represent nucleus, that's my tissue. So if I get an occlusion here, this occlusion is not going to allow 100% of blood to arrive to my tissue. They're not going to allow 100% of blood to arrive to my tissue. They're going to allow maybe only 70%. Then what about the 30% who is no longer receiving blood? They're going to die. Why? Because there's no oxidative phosphorylation. There's no aerobic respiration, no oxygen. The mitochondria, remember, the mitochondria is going to perform apoptosis, programmed cell death, to kill the cell suicide. If the cell has been damaged beyond salvage, damaged beyond repair, so the mitochondria is going to have to kill the cell, but it needs oxygen for that. There's no oxygen, so we're not going to die by apoptosis. We're going to die by necrosis, which is ischemia. Ischemia is a form of necrosis. So no oxygen, no nutrients, no ATP. These enzymes, what are they running on? They need sodium, potassium, ATPase. All these kinases, all these enzymes, they need ATP. But there's no ATP because the blood is not bringing in all the carbohydrates for glycolysis, all the lipids for beta oxidation, and all the protein for protein catabolism. That's not getting there, so we're not getting ATP, tissue is dying. In addition, why else is the tissue also dying? Besides no oxygen, besides no ATP. Why do you think the tissue will die in a stroke? There is no oxygen, there is no ATP. Also, there is no what? The third thing is missing. Yeah, there's no oxygen. That's one. No aerobic respiration. No oxygen for mitochondria for apoptosis. We cannot power energy. No ATP for enzymes. There's another thing that's missing. Circulation. Yeah, there's no circulation, no oxygen, no nutrient. But there's something else that the circulation is providing that's not there. What is it? No removal of metabolic waste building up. I'm sorry? Yeah, no removal of the buildup. You know, you can't be ordering Uber Eats and ordering pizza and you're not removing all of the garbage. So you're going to have piled up. So the same thing, this metabolic processes that oxidative phosphorylation has waste product. Oxidative phosphorylation, aerobic respiration, this glycolysis has waste, nitrogenous waste, toxic waste. So the blood also removes that. So that keeps building up. So now you have three reasons to die as tissue. No oxygen, no ATP, and no removal of toxic metabolic waste. Now the reasons are different. You have ischemic stroke, we're not getting enough oxygen, that leads to ischemia. Hemorrhagic stroke, meaning like an aneurysm. In aneurysm, You have such a high blood pressure striking the walls, making a very weak place, causing a tear. So like a balloon. You have a balloon. So in the balloon, the blood goes spinning inside the balloon, clotting inside the balloon, rupturing the balloon. The blood is now hemorrhaging inside the brain. So that's hemorrhagic stroke. Very dangerous. Okay? And so the stroke is also, the stroke is also important for, hemorrhagic stroke and ischemic stroke. And, the medulla oblongata. The medulla oblongata is part of what are the three parts that make my brain stem?
Your midbrain.
Okay, can somebody please raise their hand and tell me the three parts? Midbrains, pons, and the medulla. Excellent. Good job. Very good. 1 point. Okay. She's gonna take all the points. Before the exam, she's gonna get all the points. Pyramids. Why are pyramids so useful in our nervous system? Why are these pyramids very useful in our central nervous system? Why do we need them? Because they have the sites where I cannot hear. You know the dog can hear from very far. I'm not a dog, so I cannot hear that far. Decussations. Very good. Excellent. You get 1 point. Excuse me, that's my job. I'm the one who assigns the points. Okay? Don't tell me who to give points to. Okay, so that is our diencephalon. That is my pons. That is my medulla. It's really important to know that. It's really important to know my midbrain, my pons, my medulla oblongata. These three are my brainstem. Okay, we talked about the pons. Okay. We talked about the reticular formation. Okay, what is the usefulness of the cerebellum? The cerebellum is important for walking, movement. And the cerebellum happens to be the second largest part of the brain. After who's the largest? Cerebrum. Excellent, 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 excellent. Oh, that break was good actually. So we're gonna have more breaks now. You guys are fresh. You remember. What's the name of the fissure in the middle? You know, the same way our cerebral cortex is cut in the middle in half, so is the cerebellum. It's cut in the middle. But we don't call that the cerebral fissure. We don't call it the corpus callosum. What do we call the middle fissure that cut my cerebellum in half? Longitudinal fissure. Wrong. Midline. It is a midline. It has a name. Begins with a V. What is it? No, I don't know. What is, because it's right there. Read it. The vermis. Yeah, the vermis. So that's the middle line that's gonna slice your cerebellum in half. Don't be in a hurry to say I don't know it. Okay. You see, it looks like cauliflower. That's the cauliflower. Pickled cauliflowers are delicious.
Yeah.
Okay. Okay, if the deli man wash his hands and give you pickled cauliflowers, that's very good. Okay, the thalamus. What did we learn today about the thalamus? It is the site where what happens? We said earlier today the thalamus is important because what? It is the site. Remember when we were doing the transit steps, the first order neuron ends in the spinal cord. The second order neurons ends at the thalamus. And the third order neuron pick up the information from the thalamus from the thalamus to the brain center. Okay, so you know, like you take a trip from, you take an airplane, New York to Virginia, from Virginia to Tennessee, then from Tennessee to Texas. So you reach the top layer in your brain through the third order neuron. See, that's the thalamus. It's part of the, it is your diencephalon. The hypothalamus is very important. It has so many centers. A center for, it has so many nucleus. Nucleus is like a center. Center for thirst, center for milk ejection, center for, you see you have your ventromedial nucleus, supraoptic nucleus, the suprachiasmatic nucleus, and these are all important. Your anterior nucleus important for thirst center. Your arcuate nucleus regulate appetite, secrete releasing hormones that regulate the anterior pituitary. There is the dorsomedial nucleus, rage and other emotions. There's the mammillary body that relays, is a relay between the limbic system and the thalamus. There is a paraventricular nucleus that produces oxytocin, involved in childbirth, lactation, and orgasm. There is a posterior nucleus, functions with the central gray substance of the midbrain in emotional, cerebrovascular, and pain control. There is the preoptic nucleus, it's important for hormonal control of reproductive functions. They monitor the body temperature. The suprachiasmatic nucleus is important for, it's a biological clock. It's just beautiful if you appreciate all of these different nucleus within the hypothalamus. That's why this hypothalamus is very important, and you need to keep your brain safe, getting punched in the head. All these nuclei are gone. I mean, these guys, they're really not smart, because you're going to destroy all of these nuclei, and you're going to get a lot of money, and you're going to lose all these abilities. It's important for hormonal secretion, autonomic function, thermoregulation, and that's all shown on that figure. The cerebrum has the frontal lobe and the parietal with the bone. We talked about this in the beginning. Frontal bone, frontal lobe. Occipital bone, occipital lobe. Parietal bone, parietal lobe. Temporal bone, temporal lobes. And you know, there was in 1848, 100 years, more than 100 years ago, there was this man. He was very nice, help everybody, he's kind, he works in a factory, and by a mistake, there was some gunpowder or something, it shot up through his eyes, through his maxilla, actually here, through his maxilla, went through his eye, then it punctured his brain, went out 50 meters outside the other side. So they took him to the doctor. He actually was conscious, and he walked out to the doctor, and he said, I was hit in my brain with this, gunpowder, and, the doctor treated him for hemorrhage and, stitched up everything. And they said that he survived. He lived 12 years after, but his friends say that his emotions changed. He's cussing around, the F word all the time. He was never that kind of person. And, he's agitated. His temper changes, and he's become more negative, and, he's cannot do his job properly. So they fired him from his job. And so then when they studied his brain, they realized that they never knew before that your brain can actually control your emotion. Your brain can control your decision making. There was also a patient who had a defect in the Wernicke's area. He worked 40 years with a doctor every day asking her, who are you? But he would come to work, and he would work and everything, and she would pay him, and he would do the job finally. But in the end of the day, he would ask her, who are you, and what am I doing here, and what is this? So there's a problem known as retrograde amnesia, so you cannot remember the past, and anterograde amnesia, you cannot remember forward. So when this crisis happens, if you cannot remember the past, That's retrograde amnesia. If you cannot remember from that point and onward, that's anterograde amnesia. Okay, so stay away from the guns and the boxing, and you should be able to know what time it is today. Okay, all righty. So for your exam, for your